In this video, we'll talk about event-related designs and how they're different from block designs. So let's illustrate this briefly, what an event-related design is on the basis of this example shown here. So we have a timeline here, and we can see that we're now presenting short and discrete events with specific durations to the participants. We also have events of different classes, like faces and objects. So this experiment would look, for instance, at the bold response to faces relative to objects, and where and how these neural correlates play out in the brain. And what's also important to note is that we can now randomize the events. So we can randomize the order of the events, we can randomize the timing of the events, which is important for a number of different types of experiments. Let's, for instance, consider attention experiments where we have some stimuli being more predictive of something occurring relative to some stimuli being less predictive, such as in the Posner paradigm. There we can't have blocks. We should not present these types of experiments in the context of block designs. Also, when it comes to decisions involving value, there's a lot of value gained by looking at trial-by-trial analyses or parametric analyses, which is more difficult to implement in the context of a block or of a block design. So these experiments offer some benefits to researchers depending on what we want to study, and so it was therefore obviously researched in the early 90s whether this is something that we can do and whether this is possible within the limitations that the hemodynamic response offers, but also the scanning or the imaging environment. Another thing to notice here is this interstimulus interval, which is the interval between presenting one stimulus and the other stimulus. And it's important to leave a gap here, so basically to allow the hemodynamic response to, come back to baseline, at least almost back to baseline. So we'll talk about slow event-related designs in the beginning, which actually allow this to come back to baseline, and then later on fast event-related responses, which does not allow this to come back to baseline. So this is called the interstimulus interval, and when it's randomized in fast event-related designs, we call this jitter, this randomization. So let's consider event-related designs in a little bit more detail. So in the early 90s, it was known that the MRI scanner offered only low functional SNR, such as illustrated here in this figure, where the signal was difficult to identify from the background noise, leading to this very low signal-to-noise ratio here that we can see. So mostly block designs were used in the early days because block designs were known to increase functional SNR to about reasonable levels. So we have high detection power with block designs. But because event-related designs are important in many domains of cognitive sciences and even in neuroeconomics, people started thinking about, can we reliably measure responses to single events? Can we start using event-related designs in the context of neuroimaging? And this is exactly what Blemmer et al. Did. So they looked at visual activation to brief stimuli, but they had different durations for their brief stimuli. So basically they used visual checkerboards, which is something that is commonly encountered in the early days of neuroimaging research because it's known to lead to such a large signal increase. So what they did, though, is they varied the duration that they presented the checkerboards for from 2 to 90 seconds, ranging between 2 and 90 seconds, but also having 10-second durations and 60-second durations. Obviously, what they found for the 90-second durations are signal increases in the order of about 10%, which is quite large and nicely observable, obviously. But the question really, or the important thing for us here, considering whether event-related designs are possible, is the signal increase to single but short events, so discrete events that are shown to participants. And that's what's illustrated here in this figure. So these are short events that are presented for only 2 seconds, and you can see that there is a signal increase from about the resting signal or the baseline signal, even for these 2-second events. And this was really the first measure of the bold response to these relatively short 2-second stimuli. And the surprising thing was that even with 2-second stimuli, we could, or the researchers could observe a signal increase in the order of 4 to 5%-ish. Now, this was then followed up by a study by Savoy et al. In 1995. And they basically went a step further. They looked, well, can we look at stimuli presented below 1 second? So can we, if we present something for 34 milliseconds, is this something that we can observe? If we present something for 100 milliseconds, can we observe a bolt response to these visual stimuli in visual cortex? And as you can see from their figure here, they do observe a small response, even to 34 milliseconds. It's small, but it's observable when you average a signal, obviously. And then it gets a little bit larger for 100 millisecond stimuli, and even larger for 1,000 millisecond stimuli. So this then led researchers to propose that, well, given the fact that we can observe this, we should also be able to employ this in novel experimental designs, namely event-related designs. And so it was proposed to start with slow event-related designs. Why slow event-related designs? Well, in slow event-related designs, you basically present a short stimulus to the participant, and then you wait long enough before you present the next stimulus for the bolt signal to come back to baseline. So you would present a stimulus for, let's say, 1 second. This could be a phase stimulus. And then you would wait for, let's say, 12 to 15 seconds, maybe even 20 seconds for the baseline, for the bolt response to go back to the level, to the baseline level, before you present another response. The advantage here of this design is already that you can present different stimuli in random order, which is something that's commonly done in experiments in cognitive science and in neuroeconomics. So we can now have these designs, but it's important to understand, well, what are the differences in the activation patterns when we compare this to block designs. And this is actually what one of the earliest studies asked, which we'll talk about in a second. So we talked about why slow, because we need this bolt response to come back to baseline before we start presenting a new stimulus. And this could be anywhere in this range that we would start presenting a new stimulus. Because what would happen otherwise, if we presented stimuli in rapid succession, then we would end up saturating the bolt response. So there wouldn't be enough time for the bolt response. Let's say if we have stimuli that we alternate at 4 seconds. So we present it for 4 seconds and then we wait for 4 seconds before we present the next 4-second stimulus. We would see that we would saturate the bolt signal and it would not return back to baseline, and so therefore we would limit our detection ability. The same is true if we even presented something even faster, for instance at 2 seconds. You can see clearly that there is very strong saturation and the bolt signal simply stays at sort of the highest at its highest level. So responses quickly saturate. And this is something that Buckner and colleagues, Buckner and Bandettini and others in 1996, therefore, investigated in their first slow event-related experiment, which they compared to a block design. So let's look at what the experimenters there did, actually. So they wanted to validate the proposal, namely looking at whether block designs, which is sort of the defender, can be compared to event-related designs, which is the contender. So is it possible? Can we observe similar responses? And maybe can we even extract the hemodynamic response clearly from the slow event-related design? They used a very simple task, which is a word stem completion task. So they showed Q to participants, and they then completed this in their minds to couple, or GRE, which they completed to green. So in the block design, participants saw these word stem completion trials in a blocked fashion. So they would have a number of these followed in quick succession. Then there would be some off periods where they didn't have to complete the word stems. And then this would be followed by another word stem completion task, and so on, right? So this is a typical blocked trial paradigm. In the single trial paradigm, they would see only a single word stem completion trial, followed by some off period, followed by another single word stem completion trial, and so on. And the interval between two trials was 14 to 60 seconds long. So what did they observe here in this experiment? These are the results from the block design, which is something that we have seen now before. So in off trials, there was relatively low signal. But during word stem completion, in regions of the brain that are responsible for performing this kind of task, you'd see increased activity. And this would be in language type regions such as Broca's and Wernicke's area, that you can see that there is an increased response then, which then goes back to baseline during off trials and then ramps up again during on trials. So we can clearly see with the naked eye here that this region clearly responds to word stem completion trials and is involved in the task. Now, what did this look like in the slow event-related design, where we had these individual trials? Could we still observe similar responses there? So we had a 2-second task. So you see this, you complete this in your mind, and then there was a 40-second off period. 2-second task, 14-second off period, and so on. And it turns out that you can still see that in the same region, you see an increased responsiveness to a single trial, obviously on a somewhat lower scale. But the nice thing is, what you can do with this signal, you can then take the onset of each of these trials and average the response during these word stem completion trials, so that what you get in the end is this one hemodynamic response function that you basically then extracted from these multiple trials. So you do basically signal averaging that is time locked to the onset of the event. And this gives you this quite nice hemodynamic response. So to summarize the outcome from this study, the left image shows the activation map from the experiment, and you can see there's this right frontal area from which they also extracted, from which region they also extracted the signal then. And this is the signal from this region during the blocked trials. So you can see a clear signal increase here in the order of about 10 arbitrary units, which is quite a bit, during the on blocks relative to the off blocks. But you can also see that signal averaging works quite well within this region for single trials. So you can get a time course then, an average time course with a standard deviation around each point that you are recording to individual events, which shows a somewhat lower response in terms of the response magnitude. But what you do get is you can estimate the hemodynamic response function for this. So while the response magnitude is reduced, you can then you can observe an average response to a single event. So you have lower detection power in single trials, but you have estimation, which allows you to observe the hemodynamic response to a single trial. Comparing between these designs also showed clearly that we get similar maps. So this is the statistical map for blocked runs, which shows a lot of visual activation, but then this right prefrontal region here. And this is the statistical map for single trials for the same subject, showing also visual activation and this right frontal area that also shows activation. You can see that these are quite similar, but that there's a bit lower spatial extent in these single trial runs, in the single trial design. So basically what this showed is that slow event-related designs are feasible and can offer almost the same, detection, not quite, but sufficient detection to be feasible. But they also have one additional feature, namely that you can extract the exact hemodynamic response function to a single event.